And this is Representative Ruby's bill. He's unable to be here today, but he informed me that the individual that he put this in for is here to introduce and explain the bill. And I'm going to assume that is you.
Well, Chairman and committee again here. My name's, for the record, Dusty Backer, and let's get my ESPN here. So this bill is all about transparency. These tests that are being used do not pick up a neurodegenerative disease, because a neurodegenerative disease is just the degrading of the myelin.
Before we get started, because we are on a time constraint, how many are testifying in favor of this bill? 2? Okay. How many are opposed? Okay. We're going to set the time certain on this bill at 30 minutes for each side. In order to get going. Okay.
So just a little background about CWD, chronic wasting disease. It's actually been around since 1967. That's when it was labeled CWD, chronic wasting disease. And in 1982, Stanley Prusner came up with the theory Prion. It's a misfolded protein. And Stanley Prusner has no microscopic slides of a prion. It's a theory. To date, it's still a hypothesis. So here is a little bit about it. The group of neurodegenerative diseases. It's a TSE, which is a transmissible spongiform encephalopathy. Spongiform means holes in the brain. You can see the slide in that picture. The white is the holes in the brain. You can also see that autopsy is examined under a microscope. You have to examine the brain tissue. And the categories of TSE would be Creutzfeldt-Jakob's disease in humans, mad cow disease, scrapie, and CWD. Supposedly scrapie and CWD are the only 2 contagious neurodegenerative disease. Another CJD here, and you can see when you look up the word prions, misfolded prion, it's a lot of graphics. You're going to get a lot of graphics when you search prions. But again, there's a slide there, and you can see the holes in the brain. Now I'm going to talk a little bit about the prion theory first, and then I'll wrap it up with what the CWD and these neurodegenerative diseases really are. So here's CWD. You can see the normal brain sheath, CWD, CJD. They're identical pathology. They're both considered a prion disease, which these proteins, prion proteins, supposedly change shape and misfold, causing holes in the brain. When actually it's just the degrading of the white matter of the brain. The white matter of the brain is myelin. And myelin wraps not only the brain, but it wraps your spinal cord and the peripheral nerves. So really the deer could have Alzheimer's, could have dementia, could have MS, CJD. It's just a neurodegenerative disease. It's all degrading of the myelin. It just depends on location. Case Western Reserve, they're the leading prion institute in the nation for human cases. So all the biopsies around the United States get sent to Case Western Reserve in Cleveland, Ohio. And you can see there is believed to be a result of the change in shape. So 42 years later from when Stanley Prusner labeled the prion, it's still believed, it's still a theory. Case Western, so we got to thinking, we've been traveling around, we have no skin in the game. We've traveled the country to look for what's right about CWD because we just knew something was wrong.
Mr. Backer, how many total cases of Creutzfeldt-Jakob are diagnosed each year?
There's about 250 a year. It's about 1 to 2 per million. It's very rare, just like CWD. So we reached out to Case Western looking, we need to see a microscopic slide of this. And we emailed them, and they were actually confused, and all we asked for was the slide. So we ended up, a few emails later, a few phone calls later, we got the head pathologist administrator, and she couldn't provide a picture of a microscopic slide where all these CJD patients' biopsies go. She sends us this research paper from 2021. Allison Krauss is a big part of this, writer of this research paper. I'll get to more of that later. This one just from the CDC. I'll go through some of these slides pretty fast, not to bore everybody here. Again, CDC says believed to be a prion as a cause of AIDS. It's not is. The normal prion proteins are still not completely understood. USDA, prion protein is normal state is thought to have role and function. It's still unclear what initially causes the prion to misfold. That's why it's still a theory 57 years later. And now it brings me to the North Dakota Game and Fish. This is off their website. You click on hunting, scroll down to CWD, chronic wasting disease. And this is their backed by robust science. If you click it, the first one is the alpha document, the best management practices that the Game and Fish have mentioned that they use. In fact, all 50 states use the same document. And in this document has 162 words such as may not, potentially, suggesting, typically, unlikely. I won't read them all, but there's 162 of those words in there. Those are not science-based words. Those are consensus words. And there is no consensus in science. This is where we've gotten after 57 years. This is why we've been questioning it. 30 contributors and reviewers who wrote this alpha document. It's actually the universities and the agencies. They wrote their own document and picked out their own peer-reviewed studies. This is still what we have, is 162 words. And in their own document, page 34, it actually states there's currently no evidence that baiting, feeding, and free-range cervix can be conducted to even mitigate the disease transmission. This is in their own document that feeding doesn't even spread it. That would explain why there's no documents in the nation that it's even contagious. Again, CJD is not contagious to bodily fluids, but they're telling us CWD is. Okay, this was the third one on the Game and Fish's website. Association of Fish and Wildlife also put a statement out, and as you can say, it says indicates prions, doesn't say are prions, because again, a prion's never been seen under the microscope. And it also says that consensus, I did that one already, but there's other peer-reviewed out there with theories, including virus, bacterial, trace mineral imbalances, and others, but they chose the prion hypothesis. Okay, this is the prion hypothesis. This was the third one on our North Dakota Game and Fish website. This is their backed by robust science, the prion hypothesis. In that document, synthetic artificial prions have created a TSC-like disease. Most studies use brain material, source of prions for infection trials, which could potentially transfer other diseases and agents. However, researchers created prions in E. coli bacteria and produced disease in mice. They created synthetic prions, that is gain-of-function research. In fact, there's thousands and thousands and thousands of studies and jamming brain material and saliva and blood up these deer's nose and mouth and in their brains, and it comes down with a disease. Well, of course it's going to come down with a disease. I can't imagine the inflammatory response. And they say that since it's a protein, there's no immune response. That's because it's just the degrading of the white matter of the brain, the myelin. All the neurodegenerative diseases, there is no immune response. Alzheimer's, dementia, it's just the degrading of the myelin. This is another document, synthetic prion proteins. This is a hypothesis. This is their robust science. Is it contagious? Absolutely not. CJD is the same exact pathology as I shown. It's just the degrading of the myelin. Irrefutable evidence that CWD is contagious in bodily fluids was requested from the Game and Fish November of 23. I did not receive the study. I went through another Board of Animal Health to get the studies, and all they had was they put the steer asleep for a week, jam brain saliva in the deer's mouth, let it wake up. They did that for every week for 3 months. And they never necropsied the deer at the end. They were just tested with these tests that don't pick up a neurodegenerative disease. Again, inoculation, that is not contagious. That is not natural transmission. This is another document off the Game and Fish website. It's actually the USGS Geological Survey. It's thought to be caused by prions. There's deer on ranches in Wisconsin. In fact, they testified last session. A few of them in the room, I believe, remember. They're a positive ranch, and still not a single deer has died on their ranch. CWD, the deer are just still huge. And to date, they've only killed one because of pneumonia. This is another one off our Game and Fish website. I was going down. There's the American Veterinary Medical Association. Again, it's believed to be CWD spread from animal to animal. Still not fully understood. We've spent over $1 billion on this in 57 years, and thousands and thousands of inoculation studies, but it's still not fully understood. It's because it's a testing epidemic. And this is also on all 50 states' websites, CWD-info. This is on our Game and Fish website as well. Pneumonia is a common finding post-mortem examination of terminal CWD cases and may confuse a diagnosis if the brain is not examined. It's because you have to see the degrading of the myelin. Not on these tests, they put on a microscope and they just do staining and say they're picking up a prion. They've been testing for 40 still cannot provide a slide of microscopic prion. This is that same document, experimental infection again, inoculation. This theory is out there with minerals, viruses, nutrition. Again, there are several rare and fatal TSCs, just like CJD. CWD is no different. Diagnose the same document. Clinical signs of CWD alone are not diagnostic. Several other diseases cause similar symptoms. Diagnosis can be confirmed upon postmortem examination of the brain for spongiform lesions and accumulation of CWD-associated prion in brain and lymph tissues. The correct portion of the brain must be examined for a meaningful test. So after when we requested contagious from our department, also requested a picture of a microscopic slide, because our department works with 50 other states. In that AFA document, they have all the resources and all the people to provide a slide. Well, they sent me the same 2021 research paper Case Western sent me at 81,000 magnification. The prions are so tiny, they say. In fact, there's a presentation out there from a Creighton University 25-year prion biologist who's never seen a prion. They make synthetic prions, which is gain of function. And he said actually they're very small. They're a little bigger than the double helix. It's 81,000 magnification. So let's go through this slide real quick. The majority of it's graphics. You can see I didn't put all the pictures on here, but I did put the pictures of the slides. In the top right there, you can see it looks like kind of plaquing together. That's what they're saying are prions. But it's hard to read, and I can submit this testimony too if you all would like to read it. But when you go through the slides, A, B, C, D, E, they're all fibrils. Fibrils are just elongated connective tissue. You can see fibrils at 81,000 magnification. Prions you can't. If it's a protein, you can barely see the outline of proteins with an electron microscope. But this is electron microscopy at 81,000 magnification. And they're just fibrils. And you'll see a lot of pictures that look like this, like plaque that say these are the infectious prions. No, they're either amyloids or amyloid fibrils. Amyloids are just proteins that have plaque attached to it. That's not infectious. Amyloid fibrils are not infectious, not contagious. That's all you see other than graphics. This is the same slide in the United States that Case Western sent me, our Game of Fed sent me. And here's the test they use, and I won't read it all to you. But you can see that not once does any of these tests, the ELISA is a screening test used. The IHC is considered the gold standard because it's actually seen under the microscope. They mush up the lymph nodes and they just use some dye and then a certain staining, and then supposedly that's a prion. But again, it's still a theory hypothesis. After 57 years, would it be a hypothesis if they've seen one? No, it wouldn't. So this brings me to Dr. Joel Wallach, and I did send everybody a video, I believe, in here. I talked to everybody before. Dr. Joel Wallach has done 32,000 autopsies on over 454 different species of animals. He used an electron microscope of 126,000 magnification. The guy in Creighton said you can barely see it. It's hard to visualize a prion, this prion biologist, because it's only a little bigger than the double helix. Dr. Wallach sees the double helix with an electron microscope, and he's never seen a prion. There's 17,500 autopsies in this book alone. He's done millions and millions and millions of blood chemistries. But what's not in this book, you can about guess. There's not a prion in this book. He's seen the double helix. Dr. Wallach has sued the FDA 8 times and beat them all 8 times. The reason you don't hear about SIDS, sudden infant death syndrome, is because of Dr. Wallach. He sued the FDA to force baby formula companies to put selenium. That's why you don't hear about it anymore. They were blaming moms for laying on their babies. No, it was selenium. The babies had heart attacks. 1,700 autopsies on kids in China that had Keyshawn's disease. They were dying of heart attacks. He sued them for muscular dystrophy and cystic fibrosis, who were thought to be super genetic diseases in children. He can prevent them and reverse them, and he can use the C word because he sued the FDA. All the bears were dying across the world with liver cancer in all the zoos. It didn't matter what species it was. Dr. Wallach saved the bears. The pandas were about extinct. Some of you might remember this. They were just feeding them bamboo. That's why they weren't fertile and they weren't having babies. You can't eat just bamboo. They eat bugs and berries. And Dr. Wallach also saved the white rhino. There was 350 in the 60s. Now there's like 17,000. And And the list goes on and on and on. This book is in the Smithsonian Institute. It's $25,000. You can only buy it. No, not $25,000. We found a used one. So that's why this bill is necessary for transparency. All this information that I have in front of you today was presented in front of the Game and Fish, and I've been dismissed and told, and I quote, "You're welcome to your opinion," unquote. And that's why I'm here today. We could have this worked out, but there's only a couple individuals that sit at that table, and the sportsmen and landowners, not one. We have no skin in the game. We want to do what's right. After all the deer died of EHD, I found 150 dead deer within 2 miles of creek bottom myself. It was devastating. And the next year, they had 4 CWD meetings across the state. See, 4 CWD meetings. Actually, they added one. I believe it's 5. And it really started in 2021 when the federal funds came. Is when the real big push came across the nation. Missouri's tested 313,000 deer, and they have on 5 clinical deer. And the reason we need to see the degrading of the myelin of the brain is because in this book, the animals are dying. Every animal, every human being is dying of a nutritional deficiency disease. We keep hearing about Wyoming, Colorado, Saskatchewan. Oh my gosh, Wyoming, Colorado, Saskatchewan. Those are all just tests. A deer can have a zinc deficiency and be emaciated, spin in circles, and weigh 70 pounds, 90 pounds. It's in this book. Several nutrient deficiencies will cause these animals to be sick and lose weight. So without actually seeing the degrading of the myelin, nobody knows. And we're being restricted. Our deer are being slaughtered. Our money's being wasted. And our Game and Fish developed a CWD task force 2 years ago, and I requested to be on that task force. Casey Anderson responded to me and said, I quote, "It's an in-department task force. There's no accountability, there's no transparency, and that's what this bill is about. And I urge you to pass, and I would stand for any questions."
Representative Anderson.
Thanks, Mr. Chairman. Mr. Backer, you appear like you understand this rather well. I'm just curious. Usually when I study something, I do get a lot of information as much as I can. So how did you come about with all this data, or what made you start to be interested? I mean, did you go to school, or I'm just curious how you learned all this?
Well, as soon as, like I said, all the deer died from EHD, it became my passion. We traveled all over the nation to breeder ranches, to sportsmen's places in different states. We've worked with Texas, a big one, working with 8 different states now in the CWD federal narrative is what I call it, because that's what it is. It's just a 57-year testing epidemic. And once you start finding out what's right and what's wrong, it's very black and white, as you can see. And I just provided it. It's still a hypothesis after 57 years, $1 billion spent. Thousands and thousands of studies and inoculations. Where have we gotten? There's still 1 deer in North Dakota, and there's actually only been 1 deer in the state of Texas that was proven in court to ever have the disease by necropsy out of a 6 million deer herd.
So basically, you've just picked it up over time with your experiences.
Correct. Yep. And then we met Dr. Wallach.
Further questions for Mr. Backer? Representative Conley.
Thank you, Chair Porter. Dusty, thank you for all this. So I want to compare this to Alzheimer's. We believe it's caused by amyloid plaques. Should we quit because it's just a belief, researching it? No, we keep going and going because science changes. And hopefully we will find out if it's amyloid plaques or whatever. And I kind of see the same thing with chronic wasting disease. We can't stop studying it. We need to do the research, so would you have us stop studying it?
On CWD, absolutely, Liz. I'm sorry, Chairman Porter and Conley. Dr. Wallach, he reverses Alzheimer's, he reverses dementias. So the white matter of the brain, the myelin of the brain, is 100% cholesterol and rupture spinal cord. The deer and humans too, our livers make cholesterol. Unfortunately, we have to eat cholesterol in order to keep our brain healthy because the myelin is 100% cholesterol. If you're not taking in cholesterol, you're going to start losing your myelin. You can have Alzheimer's, dementia. It just depends on location, the brain or spinal cord deteriorating. And CWD is no different. The deer makes it in their liver, but they need 50 nutrients to do it. I don't know the exact nutrients, but they need 50 nutrients in order for its biochemistry, in order for their white matter of the brain, so they can have myelin cells, because a myelin cell might only last like a week or 2. Myelin cells don't last for months. And they need continuation of nutrients in order for their livers to make the myelin. So it's like, again, it's not contagious, because Alzheimer's is the shrinking of the brain, CJD is the whole, it's the spongy brain, dementia, frontal temporal dementia is the front of the brain, Parkinson's in the middle of the brain.
You may continue.
It's interesting. I feel like if there was a cure for Alzheimer's, we'd kind of be using it. So that's why I question some
Sure.
of this research. So thank you.
Further questions? Seeing none, thank you. Further testimony and support.